Okay, good evening, everyone. My name is Michał Burdukiewicz. I'm representing here YR Foundation, and it is my pleasure to introduce Leon Eyrich Jessen, who will today present us how deep learning can be used in immunobioinformatics. But before that, I want to firstly warmly greet all fans of R and bioinformatics from all around the world. And please write in the chat, where are you from? We are very happy to see, to learn a bit more about our viewers. And we really hope that sooner or later, you will be guests on our webinar. And before we start, I want to also mention one other thing. So today, we finally posted the application form for the YR supporting grant for women in data science, a supporting grant targeted towards exceptional female data scientists from Poland. If you are studying or working in Poland, feel free to look into the terms and conditions, apply, and we'll be very happy to support you and your growth in the Polish data science community. And in this very moment, I also want to thank thank all the people involved in the project, involving our partners from Leśniewski and Bortkiewicz company, who supported us with the terms and conditions for our grant. But coming to the man of the hour, to the Leon, I want to introduce him. So, of course, we can say a lot about his scientific achievements. Leon did his PhD in one of the most prestigious places for bioinformatics in Europe, in the Center for Biological Sequence Analysis at Denmark Technical University, the same place founded by Søren Brunak, one of the most influential bioinformaticians right now. This is amazing place, and place where exceptional people can grow and learn to do can grow and develop their skills. And Leon definitely did it. He had several postdocs, but afterwards he received his assistant professorship at DTU, where he is teaching right now. But scientific side of his work is not everything, because today he's not with us as a scientist. Of course, he will be talking about science, about bioinformatics, but Leon is here because he's very passionate about R, about data science, and about sharing the knowledge. So this is a very rare. This is not that helpful trait in academia, because academic work is not really helping you when you want to spend your time sharing your knowledge with others, sharing your passion. But Leon wants to do this. He's really great in igniting this scientific fire in his students. And also, I'm very happy that he's so willing to share his extensive knowledge. For example, I'm still using Leon's scheme of project management. You can find this. You can find the scheme on Leon's GitHub. But today, Leon will talk about deep learning in immunoinformatics, and he will show us why amino acids and why amino acid chains known as proteins can be analyzed using deep learning. So, Leon, the floor is yours.
Thank you very much, Michał, and thank you for that very nice introduction and all your kind words. I'm quite flattered. So I will just start by sharing my screen here. Like so. And Michał, can you please confirm that you can see my screen now?
I can see your screen. Everything is fine.
Thank you very much. Okay, so without further ado, I think we should get started. So the title of the talk is In Silico Immunology, Neural Networks for Modeling Molecular Interactions using TensorFlow via Keras in R. So Michał already kindly introduced me, but very briefly, I'm Leon Eyrich Jessen, and I actually have a background in wet lab molecular biology. So I used to do the lab work with pipettes and goggles and everything. But then I turned to bioinformatics and did my PhD, as Nihao said, at the Center for Biological Sequence Analysis. Currently, I'm an assistant professor at the relatively newly founded DTU Health Tech, which is a flagship at DTU right now, really interfacing with the healthcare sector in Denmark and internationally as well. I'm in the Immunoinformatics and Machine Learning group, the section for bioinformatics, and my primary area of research is how to sort of try to understand the rules governing T-cell recognition of PMHC. So I use, just briefly, I use Twitter quite a lot. I think it's a great tool for interacting with the scientific community, both within immunoinformatics, but also with the data science in general. So you can find me here on Jessen there, and I tweet about bioinformatics, immunology, machine learning, data science, RSAT, and then also teaching, which I'm quite passionate about. So first, I would like to say thank you very much for the YR for giving me this opportunity to give this webinar. And here's a picture of the Technical University where I am located. And just very briefly, for those of you who are unfamiliar with it, it's located roughly 15 kilometers, 10 miles north of Copenhagen, and the main campus covers roughly 1.3 square kilometers, which is 0.5 square miles. We have 11,500 students, 20% of those are international. We have around 6,000 employees, roughly 3,500 scientific staff, and 2,500 technical and administrative personnel. So it's a quite nice campus, and it's a quite international, and we really have an outreach to the world. So that's very nice. So here's the abstract for the talk. So very briefly, I sort of want to talk about how understanding what an immunotyping actually is important, and especially now in this time with the whole the Corona and COVID-19 situations. So I'm just going to highlight here that sort of the main keys of the talk is going to be on how to demonstrate that we can model immunotargets using these artificial neural networks that I'm talking about, how you can use R to actually use that technology. And also I will use the binding of these peptides and all the details I'll get into later to this major histocompatibility complex class 1 MSD1 as a study case. So in Silico immunology, I'll start with the basics. So let's just define that. So this is my take on general data science. You have a certain domain-specific knowledge, and then you have some mathematics background, and you have some computer science background, and you sort of work in the intersection of those three areas. In bioinformatics, we have a specialty in biology. As I said, I had a background in wet lab molecular biology. So we actually value highly that there's a strong basis in the domain-specific knowledge. In this case, that would be biology. And for this immunological bioinformatics, immunoinformatics, the particular area of biology that we are using is immunology. And this is equivalent within Silico immunology, basically. So I would sort of frame it as the study of the immune system by mathematical modeling implemented using programming. So it's really intrinsically interdisciplinary, and it's important that you understand all the areas and you're able to connect them. They'll sort of give you the sum of 1 plus 1 plus 1 will be more than 3, and that's sort of the basis of this way of thinking. Now that we defined that, I would like to continue talking about artificial neural networks, and I have deliberately spent quite a bit of slides actually explaining the basics, and I have done so hopefully in a way that anyone can be part of this talk. I've really tried to be explicit about that no prerequisites were required, so I really hope that it's possible to follow the following. So an artificial neural network, it's a mathematical framework for mimicking how the brain processes data. So here on the right, we have a sort of a cartoon sketch of a brain cell. We have one cell here, never mind the labels, these are not important currently. And then you have some interactions coming from other cells. And that there's the primary sort of interaction propagating from the cell. So the human cell actually consists of roughly 100 billion cells. And together they form this intricate network where most are connected with most and able to communicate via actually electrical signals. So through hundreds of thousands of years, literally, evolution has really fine-tuned our brain for this one purpose. We need to be able to process massive amounts of data, and we need to do it in real time. We even need to sort of sort out in the data. And our brain is actually able to process vision, smell, taste, sound, physical touch, emotions, all of these, and that this is really done for you to exist, interact with the world. And all of the above is happening to you right now. This means that right now your focus is hopefully on my voice talking on this presentation, but actually your brain is registering a lot of stuff around you right now. So some of it you will not react on, you'll just ignore. But trust me, if you smelled smoke, for example, you would immediately identify that. So your brain is really able to process the information and find the essential for you right now. So if you think about it, it's quite cool. Our brain is actually a data center. It's quite amazing how biology works and how nature is able to optimize solutions. So moreover, how do we actually get from a brain cell to an artificial neural network? So if we take this figure that I showed you before as a point of reference, this brain cell cartoon, then we can remove the labels, and then we can add these stylized neurons. So basically just showing each of the brain cells as one neuron. And then we can also add these directions as forming a graph. So note that the arrows have directions. A signal is coming from one cell to another and only in that direction. So if we remove the brain cell, we sort of get a stylized schematic of a neural network. So we can just rearrange these neurons so it looks more natural. And that would be sort of gathering the nodes here. And on the left, a lot of nodes have connections to one neuron. And then this neuron has a connection to 3 neurons. So of course you would have more neurons in the middle connected, and more will be connected in general. So with respect to general, that's generalize and simplify. So here we really have sort of the basic schematic of a neural network. You have some neurons here, and all of these are connected to each other. So this neuron here is connected to all of the next layer of neurons, and likewise for this. And then they have this layer here, which is then connected to the final layer. And now I'm using the term layer, so let's move on. We have an input layer here. So this is the input to the neural network. Then we have a hidden layer here, and there can be multiple of those, hence the S in parentheses. And then we have the output layer. So that's the basic schematic: an input layer, one or more hidden layers, and an output layer. So each of the edges here, the arrows, that's a weight, which will become a parameter in network. I'll explain that. And each node is basically a calculation over the weights and inputs. And finally, each node has an input value, meaning that something comes in to the neuron, and an output value, meaning that something goes out from the neuron. So how does the ANN actually process these input? If we look closer, we have the weights that I talked about before. Remember the arrows, the edges are weights, so we have inputs here. And we have weights. Each weight is associated with its own input. So basically, it's the input to this neuron here is this value times this value plus this value times this value plus this value times this value, and so on. So this will look like this. You can see here on the right side that the input here is the sum of the products here. And this is exactly the same for the next neuron. Now we just have some new weights. So note that the weight connecting these 2 neurons here is not the same as the weight connecting here. These are different. And remember, these will become parameters in the network. And for the final one here, this is exactly the same. So now we know how to calculate all the values for the hidden layers 1, 2, and 3 here. It basically looks like this. It's exactly the same. So we can sort of put the calculations here together. And we can see now that these 3 values is the same. These 3 values are the same. These 3 values are the same, and so on. So let's do like this instead. Now we have a set of weights. Remember, these were different from these weights and were different from these weights, and so on. But let's put the weights together, and let's put the input vector together as well. Then we ended up something like this. And if you've touched a bit upon linear algebra, this might look familiar. Actually, what we end out with is that the vector here of the hidden layer is the matrix multiplication between the weight matrix and the input vector. And this is really, if you're wondering why everybody's talking about GPUs in deep learning, This is the reason. Calculating the value hj is independent of calculating the value of hj plus 1. This means that it's very easy. It's called embarrassingly parallelizable. So we can basically just do each of these calculations in parallel, and this can really be sped up by GPUs which are specialized in this. And we can even expand this to take multiple inputs at one, pass through the weights, and get multiple hidden layer values here. So now we really have matrix multiplication, and this is exactly the basics for why GPUs work so well, and the basis of neural networks. It's linear algebra, it's matrix multiplication. Okay, so actually it looks like this if we use the matrix, the algebra notation. So basically it boils down to this, and for the output, it's exactly the same. But there was this thing about nonlinearity, and what I showed you before, the matrix multiplication, that's just a series of linear combinations. But remember also that I told you that each input neuron had an input and an output. This actually means that the input to this neuron, for example, will be the matrix multiplication between the weights and the input vector here, but the output will be passed through a function. And this really relates to how a brain cell works. As I told you earlier, you actually get electrical signals into the neuron here. And then if these signals, the sum of these signals, if they are large enough, then one signal is passed on via this axon. If they're not large enough, nothing happens. Okay, so that's the biology, and we can use math to describe that. Basically, we can use the activation function. If you work with deep learning or neural networks in general, you've encountered this notion of the activation function. And in this case, you can see that it's 0 if x is less than 0. Just as I told you before, nothing happens if it's too small. And it's 1 if x is larger than 0, then a signal is propagated. So there are different activation functions. One could be, for example, the leaky ReLU here. Let's actually talk about the other way around. Let's take the step function here first. So it basically just jumps one step around 0. So it can only be defined between 0 and 1. Another popular one here is the sigmoid, which is also only defined between 0 and 1, but has a more sort of a gradual incline from the 0 value to the 1. And then there's this ReLU, which is being used a lot, which is actually quite simple. It's just 0 if x is smaller than 0. Otherwise, it simply just propagates the signal. And then finally, there's the leaky ReLU here, which has a slight slope if x is less than 0. Otherwise, it just propagates the signal. And you can see sort of a notation of the functions here using pseudo-code and R functions. So that's the activation function if you've encountered that. So why do we want to use artificial neural language for this problem? What's the deal? Well, basically, if you look at this word really quickly, don't think about it, then I'm pretty sure that you will read human. I've used this figure many times in talks, and people do actually see the human straight away. But if you take a closer look, what is actually there, it's human. So it's HUMHN, right? But we can easily read human. We can actually easily read it here. We know what it's supposed to be, right? And that's really our brain's ability to capture the context. So it's really it's able to know that if we see the H first here next to a U, it should be an H. But if it's squeezed in between an M and an N here, we should interpret it actually as an A. So not only the value of the variable matters, but also the context in which it was observed matters. Okay, so that's really the nonlinearity, and this is what you get by this nonlinear activation functions. Okay, so that was the artificial neural networks basics. So now you know that it's quote unquote just linear algebra, and then you have these nonlinear activation functions. So let's talk about a bit about the immunology. Remember the 3 circles I showed you about understanding the different areas that we're supposed to, or we're going to combine. So the basic life of your body is a cell, and it's defined sort of by having an outside, you have a barrier, and then you have the inside of the cell here. And inside the cell, you have proteins. Proteins are sort of the active part of the cell, structural and also sort of in functions and tasks that needs to be carried out. And these proteins, if they were just constitutively constantly produced, They originate from the DNA, by the way. But if they were just produced constantly and nothing else, the cell would simply clutter up. So they need to be broken down again once they sort of serve their purpose, okay? So the protein is captured by this proteasome, which then breaks down the protein into peptides. These peptides, they're nothing more than smaller fragments of the protein. And there's a real note here we need just to touch on it very briefly, we'll get back to it later, that peptides are instead made of these biochemical building blocks that we call amino acids. And there are actually only 20 of those in your body. And you should just basically think of Lego bricks. If you have sufficient diversity in your Lego bricks, you can almost build anything. And that's the same principle with these amino acids. So full proteins broken down to smaller stretches is called peptides. And the individual components of the peptides, which is then makes up the proteins, are called amino acids. We'll get back to this later. So a fraction of these peptides, they're bound to the MHC molecule, and then they're presented to the outside. You can see here the MHC is here, and the peptide is here, and these are going through this pathway here and ending up on the surface of the cell where they're accessible to the outside. So this T cell, which is a specialized immune cell, can then read these peptides. And if the cell is sick, the T cell actually has the capability to kill the cell. And this is actually really, really important. But how does it work? Well, under normal circumstances, the proteins that are produced inside the cell comes from itself, the DNA that's in the cell ends up as proteins, basically. But in the case where you have a virus infection, the virus will take over sort of the machinery of the cell and start viral proteins will begin to be produced. These are also broken down. This means now that we have a breakdown of these as well, we end up with peptides that actually are not from inside the cell itself, but peptides that originate from virus proteins. So this is non-self peptides, but they also go through this machinery, this pathway, and end up on the surface here. Now the T cell is actually able to recognize that this is now no longer a self-peptide, it's a non-self peptide. And that's really important because what happens then is that the T cell is able to induce programmed cell death. So basically the T cell will kill via a signal sent the cells that are infected. And that's really important. And if we can understand what the rules that are govern this interaction, but it's a molecular interaction, right? You have your MHC molecule, you have your peptide ligand here. They bind together. In order for a T cell to get activated, a non-self peptide must be recognized. So if we can understand what makes a non-self peptide and whether that will actually bind to the MHC, then we can get a lot further in actually understanding what's going on a molecular level. And that can actually be used to create very specialized targeted vaccines. So if a peptide is bound to the MHC and recognized by the T cell, but then we call the peptide an epitope, okay? So an epitope is a peptide capable of activating the T cell. And this is actually exactly how our body fights COVID-19. So if you've been sick from corona, then you've had virus particles inside your cells, and then you've had your immune cells seek out your sick cells and basically kill them, okay? Very briefly here, this is not the entire story. I'm leaving out some details, and you've probably heard about antibodies and antibody tests and so on and so forth in order to sort of show if you've had a coronavirus infection. That's also really important, but that's another topic for another webinar. For now, we will focus on this T cell part. Okay, so let's tie it together and talk about sort of this molecular machine learning mathematical model. So how do you use math in the context of creating machine learning models to model molecular interactions? So we want to create predictive models. We want to be able to predict sort of the primary task of most machine learning models is to predict. So we have a model, we use it to predict what's going on, what's likely going to happen in the future, or in this case, if two molecules are going to interact or not. So the key task of this predictive modeling is really to capture and quantify the transformation of input to output. Okay, so remember we had input vector and we had some output, and then something happened in the middle. This something is a transformation, right? You have the input coming from a high dimensional space, and then you want to basically map it down to just a single value via this transformation. And that transformation is a mathematical transformation. Okay, so let's see it in context with this MHC class 1 and the peptide here. So the same system as before. The blue one here, that's the MHC molecule, and the green one here is the peptide. So now I want to know, if we put this MHC together with this peptide, what will then happen? So we want to know what's going to happen. To do that, we need to know the amino acid sequence of the MHC molecule, and we need to know the amino acid sequence of the peptide. Luckily, we can get these, again, skipping some details, but it's possible to get the exact amino acid sequence of both the MHC and the peptide. And in that, the system is actually quite confined and closed, and the peptide really sort of clicks into the class 1. Then the information on whether or not they will bind must be present in the amino acids, which define the biochemical local environment. So we just, quote unquote, need to find the transformation of these amino acid sequences, and then they will tell us whether a given peptide will bind to the MHC or not. Good. Let's take a closer look at this. So let's map it on to what we were talking about before. We have the MHC and we have the peptide. Put them together. What will happen? This is the input. The transformation is here, what's going to happen, and the output is, are they actually going to bind on it? Okay. Again, recall we discussed ANN. They take some input, performs a transformation, and gives some output. So this is sort of the mapping of our molecular problem here to a more generalized form. So let's see if we can actually, as we talked about before, capture and quantify the transformation. So what we do is that we need some training data. And training data is really when you know the input and you know the output. So how do we get that? Luckily, a lot of data is available online, and luckily we have very good collaborations with people who are extremely skilled in the laboratory. So they can go into the lab and say, given this MHC molecule and this peptide, will they bind or not? So that's the training data. We have input and we have output that's known. The key is here, we know what it's supposed to be. So basically, we know the input here. It's the amino acid sequence of the peptide and the MHC. We don't know the transformation, but we know the output. Okay, so that's the setup. So how do we actually capture and quantify this unknown transformation? What we do is that we feed the model the first training data point, and then we perform a random transformation. And this is literally random. All the weights that actually perform the transformation are just initialized random. So the first stuff that will come out of this is actually complete gibberish, but something does come out, okay? This fact that something does come out, we can then compare that to the known output that we know, the target value. We can quantify what is the difference here. And from that, we can calculate an error. If we calculate this error, then we can use that to slightly update the transformation, which will then reduce the error, okay? Then we repeat that exact process, and we do that once more, and again, and we do that once more, and again, and again, and you get the picture now. We keep doing that, and then at some point, we will actually have a model which is capable, hopefully, of accurately reproducing the actual known output, the target value, okay? And if we get that, then we really have our predictive model. So very briefly on sort of the main output of the group that I am an assistant professor in are these two predictors. So if anyone is online listening and knowing about this area, then most likely you've heard about NetMSC PAN and NetMSC 2PAN. These two servers, which are available at these addresses that I've put down here, have been trained on roughly 13 million data points covering roughly 250 different versions of this MHC. And for the class 2, some details here I'm going to leave out, but it's 4,116,000. So it works really quite amazingly. This works better than this one, but both are quite capable. So you take the MHC, you take the peptide, you perform an encoding, we get back to that, use it as input, feed it through the model, and then you get your output, which is then the prediction, which is then based on these huge data sets. Moving on, how do we actually do the conversion of biology to numbers? So I showed you this encoding block here, and now we need to talk about the 20 amino acids that I talked about earlier, the Lego blocks. Remember, for all proteins in the human body, you are produced by just these 20 amino acids. It's really quite amazing. And we can note them down schematically, cartoonishly, I guess, like so. So 4 times 5 here, where each of these have a biochemical profile. Some are acidic, meaning negatively charged. Some are basic, meaning that they're positively charged. Some are hydrophobic. This means that they don't like water. Some are neutral. They don't really care. And then there are the polar ones here, and they do like water, actually. This is sort of the color of the general classification of the 20 different amino acids. But even though they are both red, like these two, for example, they are still different. In this case, this one has a slightly larger, this one is slightly larger than this one, for example. Each of these amino acids has their own unique biochemical profile. So if we circle back to this human that I showed you, where you were able to interpret this as an A, this is really why we need the artificial neural networks. If we look at this example in the context of actually producing a small peptide, remember peptides were small stretches of amino acids, then we can have a positive here, for example, a neutral, a negative, a positive, and a negative. Note that the R here is the same as this one. At least that's what it looks like. The first R is the first amino acid, first of all, and is only flanked by one amino acid, which is neutral. The second one here is sitting within the peptide, and it's flanked by 2 negative amino acids. So if you think about it in the local biochemical environment, the first R is simply not the same as the second R. Context really does matter, and this is exactly why neural networks, they excel at this particular task. So the MHC up here, that's 180 amino acids. And the peptides here, they are somewhere in between 8 to 11. Most predominantly, by far, they are 9 amino acids long. And that's what we will work with later in this webinar. So basically, it could look like this. You have a Q at the first position, a D at the second, and a lot of stuff in between. And then you end up with an I in position 180 here. And here you have Y on the first position, L on the second. A lot of stuff goes on. Actually, not that much, just a couple until we get to the 9th position here, which is then a methionine. So how do we actually do the encoding? So if you unfold the matrix I showed you before to just sort of be one vector against another vector, all against all, you can say. But then we can use ways of saying how similar are 2 amino acids. So in this case, for example, we say, well, we need some similarity metric for L here and I, and that similarity matrix basically quantifies how similar, biochemically speaking, are L and I. L and I are actually quite similar, so in this case, X will be quite high. But if we take E here, you can see that it's red, positive, and H here, it's blue, it's negative. They're quite dissimilar. So X is low, okay? And then you just need to imagine that all of these values are here in all this 20 by 20, so 400 values in here. So how do we actually do the encoding? We do the encoding, we wanted to encode this lysine here, the K. We would take the 20 values of the similarity to alanine and the similarity to arginine and so on and so forth, and then we'll get 20 values describing this particular amino acid. So each single amino acid is basically encoded as a vector of these 20 similarity scores, okay? This way, we are able to actually map the distinct biochemical profile of each of these amino acids to represent them as 20 values in the amino acid space. That's how the encoding works. So remember that we had 180 positions here, 180 amino acids on MHC. And now we need to convert that to input. This means that we would need 20 values for each amino acid. There's 180. This means that we need 3,600 input values. In the case for the peptide, input values here, there are just 9 predominantly. This is what we can work with. So that's 180 input values. So 20 values for each amino acid yields 180 input values, means that there's 180 elements in this particular vector here. So in the following example, we'll work with one particular MHC type, the HLA-A0201, and 3 classes of 9-mer peptides. We'll call them strong binders, meaning that the interaction between the MHC and the peptide, the molecular interaction is strong. And then there's the in-between, which is a weak interaction, and then sort of the non-interaction. Those are the 3 classes we'll use for this example. And then we need to remember that in this case, I just said that the MHC here, it's the same for all peptides. This means that there's no reason to include this in our model because that would just be constant. So we're just going to use 180 input values for our network. And then the output here will be, does this peptide actually bind to the MHC? Yes, no. And this will then encode as 1 and 0. And then there's a score in between, sort of quantifying whether or not they will bind to each other. Okay, so in summary here, I said I would demonstrate how we can model immune targets using artificial neural networks. I will actually get to that now, but I hope I sort of was able to go over how to use this binding of peptides to the major histocompatibility complex as a study case. So just an overall summary of what we've been talking about in the first part of this session. In Silico immunology, that's the intersection of immunology, mathematics, and computer science. Artificial neural networks, these are mathematical frameworks inspired by the human brain and quite capable of handling this information in the nonlinear matter. Remember, we basically just had linear combination, matrix multiplication, and then these nonlinear activation functions propagating the signal between the neurons in the network. And then it's important to remember that this presentation of a peptide to a T cell is a crucial step in immune activation. And this is really what plays a big role when the cells get infected by viruses, which are capable of penetrating and get into the cell and make a lot of mess in there, so to speak. And with respect to how we actually get the model to work, we need data where we have known input and known output. So this is training data, and we use this to train an artificial neural network to predict whether or not these 2 molecules, the peptide and the MHC, are actually going to interact. Finally, remember that proteins and peptides consist of amino acids, and they can be converted to a numerical representation using these similarity profile vectors that I showed you before. Okay, so let's move on to TensorFlow via Keras in R. So what you need to know about Keras in R. I'm not going to go into like huge details because there's an entire book called Deep Learning in R, which is a really good book, which I would recommend. But what I think you should know is that it was originally developed by research and engineers working on this Google Brain team, where they did all of the machine learning. So how do you actually get a really efficient search algorithm? Or if you're using Gmail, then you see this autocomplete, which is really quite cool. And then they decided at some point that instead of just keeping it in-house, it should be available to the world. And then they open sourced TensorFlow. And this R version of TensorFlow, that was officially announced in February 2018 by J.J. Allaire, who's the CEO of RStudio, at his keynote at the RStudio conference. And as I said, TensorFlow is an open source library, and they can actually be used to do general numerical computations. So it's not just confined to doing neural networks. You can do any computation. So if you need to do faster implementations of calculations, then you can access this numerical computation API, which is written in C++. And actually, the core TensorFlow library is in C++, but you can access it via Python. And now comes the tricky part, because on top of TensorFlow API in Python, we have this Keras. And what Keras is that it's a high-level neural network API, and it's also written in Python, actually. So Keras was originally developed by Francois Choulet, who's working in Google, because he would like to have an even higher level framework than the Python API to TensorFlow in order to really be able to go fast from ideas to experimentation. I really like this quote about being able to go from idea to result with the least possible delay as a key to doing really good research. Okay, so in summary, the Keras R API calls the Keras Python API, which calls the core Python TensorFlow API, which again calls the TensorFlow C++. So there's quite a call stack going on. It's a bit complicated, granted, but in my experience, for the most part, it seems to work quite nicely. Yeah, and then just to highlight that this is a really key if you are spending all your time on doing really complicated implementation in C++. I mean that can be fun, I would agree, but it's a hindrance in you moving forth in the application of these tools. Okay, so let us do some coding. So what I'm going to do now is that I'm going to stop sharing my PowerPoint screen here, and then I'm going to switch one moment here to my let's just see here, my Google Chrome. So Michael, I assume that you're still online. Could you just confirm that you can see my RStudio Cloud workspace here?
Yes, Leon, I can.
Thank you very much.
It's opening.
Excellent. So there's just a short load time. So I took the chance of doing this on RStudio Cloud, and the reason for that is basically that I want to, when I'm done with this, I want to open this particular workspace to the public, and then I'll send the link to the YR Foundation, and then they can put that in the description of the video, so you can access it after the talk. So what I did here is that I've just made a small example, and I'll just go through that code line by line. My apologies, I was not brave enough to live code all of this. I hope you can live with that. So basically, the header for our first script here is that this is a scaled down version of a blog post on how to use TensorFlow for R that I have on RStudio. That was quite amazing. I was contacted by JJ Allard, our CEO of RStudio, and invited to do this blog post. And actually, a word of advice that was initiated on the basis of me having shared some of my teaching code on GitHub, which he then found interesting. So that was quite cool. So the aim of what we're going to go through now is that we want to create a predictive model which is capable of predicting if a given peptide is a non-binder, a weak binder, or a strong binder, as we talked about. So basically, this is an artificial neural network-based three-class classifier. So what I'm going to start with here is that I'm going to start by clearing Workspace. I'm very sorry, Jenny, for those of you who know what that refers to. But basically, I have to do it because I only have 1 GB of RAM, and I can very quickly end up in problems if I don't think about that. So let's start by clearing the Workspace like so. Then we want to load our libraries. I'm a keen user of Tidyverse, which I would recommend you look into if you're interested in sort of a modern way of doing data science. And then I have a function file here. Inside that function file, I can just show you here. This is written in base R, so it's a bit more cumbersome to read. But basically what it does is that it takes a peptide that we discussed about before, and then it does this encoding, creating a vector with 180 values. Remember, we had 20 values that we needed for using the amino acids, 9 amino acids, 20 encoding values, 180. So this is basically just what this function does. So I call that into the workspace using the source here. And then I want to load my peptide data. I made a small data README here. So basically the data consists of a total of 23,760 data points, and it has 3 balanced classes, each with 7,920 data points. And then, as we've talked about for the past 45 minutes roughly, knowing how to identify an epitope is really paramount in vaccinology. A peptide may be an epitope if it's a strong binder, less likely if it's a weak binder, and not if it's a non-binder. And again, I'm skipping some details here to keep it within the limits of what's possible on a webinar. But basically, MHC presentation is a requirement for creating an epitope, remember epitopes were really defined by activating the T cell. So the final step is on the T cell side. So the data that we're using here is actually in silico data. So this is a set of random peptides where I have predicted whether they are binders or not. So technically we're actually modeling a model, but since NetMHC-PAN works as well as it does, it is not far from having real data, quote unquote. So the variables in the data set is the peptide, the amino acid sequence of this 9 mers, and it has a label whether they are non-binders, weak binders, or strong binders. So let's just briefly view that data. So it looks like this. We have the peptides here. Remember, we have these letters L, L, T, D, A, etc. And then we have a label here. Then we can see the class distribution, 7,920, as I showed you before. So now what we want to do is that now we want to convert this into having numerical representation for the labels, like so. So this is just taking strong binders, saying these are 2, weak binders, these are 1, and non-binders, these are 0, as seen here with this case when function. So now we want to define some training and some test set. So the data that we read in here is just all of the data. So we just need to split it. So I'm setting a seed because I'm doing random sampling, and then I'm assigning 20% test and 80% training data. So that ends up looking like so. Now we have the peptide, we have the character label, and then a numerical label, and then we have an assignment. For example, this particular data point ended up in the test session. This means that we will use all the data points here that are designated train to train the model. And then once the model is trained, we will see how well is it doing on the test set. It's very important. The test set is not part of the training in any way. So let's see how the random sampling ended out. We can see for the non-binders, roughly 1,500 and 6,000 test train, and this goes on roughly for the others. So that's quite nice. So for actually doing the conversion, this similarity thing I was talking about, if you want to know more about that, I added a few links here that you can go read about, but basically it's called a blossom, and in this case it's the 62 version of the blossom. It looks like so. Remember I told you that you had all of the 20 amino acids I showed you, and then you have 400 values here. So we have A and 20 values here. Each amino acid will be encoded as these 20 values. So if it was ARN, for example, it would be a vector with these 20 values first, then these 20 values, and then these 20 values, 16 in total. Again, remember we have 9 times 20, so that's 180. Okay, now we need to define our training and test matrices, and we do that by taking the peptide here, and then we filter for the training set. Then we pull out just the peptide, and then we use this encoding function that I showed you with the blossom as the encoding matrix. So let's just run that, and this one as well. And we also need to define our Y values. So these are the targets. Remember whether or not they will bind. In this case, these are actually class targets. So it's a matter of whether they are 0, which are non-binders, 1, weak binders, and 2, which are strong binders. So let's run this code, and then, as always, inspect the data. So it just stopped briefly. Let's hope that it kicks on soon here. It's always a bit when you use cloud services. I do have a local version of this lying around. Okay, so we got some warnings here. In my earlier test of this today, that made no difference. So let's just press Control L and ignore those. Let's see how it turned out. So here you can see the encoding matrix of the first 6 peptides. So LL, TD, so on and so forth. So the first values here, next values, and so on and so forth. And let's actually also just check the dimensions so we are absolutely sure that we agree on what's going on here, like so. And we can see that we have 19,000 peptides, and then we have these 180 values that I keep talking about. Rows, column, 180 values per peptide. Okay, the test looks like the same, but let's look at the Y. That's a little trick to this. This is actually being converted into a categorical representation using what's called one-hot encoding. This basically means that all the three classes are now orthogonal. They are in a right angle to each other, 90-degree angle if you were looking at them as vectors. But basically it's just a matter of saying that the first peptide here, that's class 1, and then this is a class 2, class 1, class 1, class 0, class 2, etc. So that's just the way that this works. And if we map that back to here, let's see what we were looking at here. This was the training data for the Y, meaning that we can see that the first peptide here, that's a weak binder. So that's 1 here. Corresponding to that, there's a 1 in 0, 1, 2 in this column here. Second is a strong binder in the training set, so that should be over here, which is, okay, that looks like it actually works. Okay, so what we need to do now is we need to set the parameters, the hyperparameters for the neural networks. And hyperparameters they're really important, and they're a huge topic of research and how you get the best set of hyperparameters. This is what I'm always asked. How do you get the best hyperparameters? And unfortunately, there's no answer. Let's start by setting these hyperparameters. We just do that as variables in the environment. Just going to run through them like so. See here how we have the number of hidden neurons in the first layer. It's 180. Second, it's 90. Then it's 45. Then it's 20, and so on. We have the activation function we talked about here. ReLU we're using in this case, I can see. And then we have this dropout thing. So dropout is a way of preventing overfitting. And overfitting is something you should be super aware of when you're doing neural networks. There's so many parameters you can easily overfit. Overfitting means that you are modeling your particular data, including the error that's in the data, and not the general trend. And what you really want to do is you want to model the general trend. This dropout basically takes each of the weights, and then it says, well, there's actually only a chance that you're going to get updated. In this case, with a 40% chance, you're not going to get updated. On the second layer, it's 30, 20, and so on and so forth. For the final output here, we're using softmax. Remember, we had 3 outputs. So we have 3 classes, and we have multi-class classification. We use this softmax. And then we have a number of epochs. One epoch is one run through of all the data, just some terminology here. And in this case, the batch size that I'm highlighting here, that's how many data points you're taking in before you're calculating the error. The idea being that if you have, for example, a data point, which is sort of a bit odd, a bit outlier, then the impact on the weights of that might be quite substantial. But if you take more data points in at one and then do it over the average error, then you are more likely to sort of correct your weights in the general direction of the error. And then for the loss function, this is the one that is used to define the difference between what we are predicting or what we're supposed to predict. In this case, where we have 3 classes, we're using the categorical cross entropy. The learning rate here, terminology, that's just how big a step should you take each time you update the weights. Okay, so this network can be a bit difficult to sort of get your head around. So I just want to show you something that I did just for fun. Let's see if that is up and running. The shiny apps I just need to wake up. So basically I did this small dynamic visualization of architectures. It can actually be quite nice if you're working with the big networks to get an idea of what is a big network and how does it look. So in this case, we actually have 180 input neurons. We have 3 output neurons. Let's make the neurons slightly smaller. And then we need to define the hidden layers here. So we're just going to back here and we have 90, 45, and 20. 90, 45, and 20 before we hit the output layer. So let's put that in here. So it's 90, 45, and 20, right? So basically it looks like this. And now you can sort of get a feeling for how many parameters are actually in a neural network. Remember, this one node is connected to all of these nodes. And that's the case for all of them. And remember, this one is connected to all of these nodes, etc., and etc. This means that each of these weights is a parameter that you can train, and you can see this network, which actually sort of kind of looks like the Eiffel Tower, and I'm thinking about it on the side, contains a lot of weights, so there's a lot of parameters. Okay, so just wanted to show you that. Let's close that down again and return to the RStudio Cloud here. So we ran all the hyperparameters. Now comes the definition of our model. So what we're going to do here is that we're going to start with a sequential model. This just means that we are able to build the model in layers. The first layer is a dense layer. It has a number of hidden neurons, one which we said earlier on, that was 180. It uses whatever we define as activation function, that was ReLU, and it receives the input of 180. And then it goes through a dropout layer, and then this is repeated several times, as I showed you in the Eiffel Tower, basically, until we end at the end where we had this output layer here. So let's set this model like so. And here, the next step that we're going to do is, again, these errors seem to be ignorable, so let's cross our fingers. We're going to compile the model. Here's a little quirk that you're going to have to notice. There's no assignment, but calling the compile function on the model object actually updates the object. So this is really object-oriented, right? Compile the model like so. And now we can actually get this quite nice summary of the model by calling the summary function, lo and behold. Now we can actually see how the layers look, 180, how many parameters are in each of these layers, and how many parameters are there in total. So we see here roughly 55,000 parameters are here in total. Now we will train the model, and we'll do that by calling the fit function on the model object, which will return some history, but it will actually also show us dynamically the learning, the training of the model. So now we're running an epoch. Remember, one epoch is running through all the samples. And we can see down here we have the loss function and the validation loss. And what this means is that I set the validation split to 0.2. This means that each batch it takes in is 50 data points. It will then train the model using the 40 data points and then sort of make a temporary online evaluation of that particular data point on 10 that is not using for training. And this is what we can see here. We can also see how the accuracy and the validation accuracy are sort of actually converging over here. So one epoch, 2, 4, 6, 8, runs through all of the training data, run over the data several times. And here on the y-axis, we have accuracy. So how many do you get right out of how many you actually predicted? Okay. So what we're going to do now is that now we're going to evaluate the model. I'm just going to run some performance metrics here, create something we can use for plotting. And then finally, I will plot the visualization of the predictions that we did. So let's see if we can pop this out. So what you can see is here, we have the test pane here. So remember, these are the data points that the model has not been trained on. And there are training pane here. These are the models that the data points that the model has actually been trained on. If we're doing well, we should be in the diagonal, which we are predominantly. And we can see that the training accuracy is roughly 96%, and the test accuracy is roughly 93%. This you will see often, and it's an indication of a slight overfitting here. Dealing with overfitting is unfortunately beyond the scope of what we can go through today. But what I want to show is how easy it actually is to change your setup. Let's say that what you actually wanted, let's say, I don't want the dropout layer, and maybe I just want the 2 first layers, and then I want to skip the rest like so, and then go directly to the output. Okay, let's see what happens then. Then we create the model again. We recompile the model. We get a new summary of the model. Now we can see that there are less parameters. It's still quite a lot. Maybe we should reduce that further. Then we can go up here and just say, let's pull out this layer as well. Let's see how then that looks. Compile the model, see the summary. Okay, so now we're down to roughly half the parameters of what we had before. Let's train the model. It looks just like we did before. So you see it running over the epochs, running through the data. It's a bit quicker now because there are less parameters in the model. Now you can see, if you noted, I took out all the dropout layers, which I told you were used to handle overfitting. And you can actually see exactly what is going on here. You can see that at this point, you sort of get a quite nice generalizability, but then you actually end up going sort of more and more closer to your training data and more away from your actual validation data. Okay, so this is exactly overfitting as I talked before. Just to create the plotting stuff that we need, run the plot, and now we can see that it looks like this. So roughly 97% accuracy on the training and roughly 92% accuracy on the test. It's still quite all right. So now we're going to save the model that we created, like so. It's very easy. And here, this is really cool because this model object here, H5 file format, can actually be read directly into Python, which means that you can create your models in R and then move them on to your colleague, which is where R is working in Python. Okay, so that was actually the first part of showing you how you can use this. But now to an important point, what went on, what was actually learned by the model. In order to look into this, we can use what is called post hoc analysis. And again, I'm going to clear the workspace. Sorry, Jenny. And then I'm going to run the library commands that we need, source in the project files, and then this updated peptide file, like so. And then I'm loading the model here. Simply just load model in, and now I have the model back in a new script, and I can actually use this model for something. So let's add the predictions. So take the peptide and make a mutate, and then say, okay, I want to add predictions on Y. This peptide need to encode it first, and then just into the predict classes using PMHC model object here, and then the encoded peptides. So that is like so, and this means that now the data looks like so, meaning that we have a predicted value here and a numerical value here. So what we want to do is now we want to look at, let's see here. We actually need to add something here. I can see I forgot. So let's just look at the training set. Oh, sorry. Training, like so. So now we're looking at the training set, which should of course be the test set, sorry. The test set, was not used for training the model, so that's completely independent. This means that let's take all the data points that were predicted to be strong binders by the model, and then see if there's information in this. There is. Again, here we're taking all the peptides that were identified as strong binders by the model, and only looking at the test set. So this is a post hoc analysis, including this here. We can see that it seems that the peptide position, so this is the first amino acid in the peptide, second, third, fourth, all the way up to 9. It seems that the second and the 9th position harbors a lot of information. These are bits of information. This is a Shannon logo if you're interested. So let's compare with how distinct the information from the second position and the 9th positions are between the strong binders and the weak binders. Now you can see the information is less in the weak binders, and let's see how it looks in the non-binders. Here it disappears completely. Okay, so this is a way to decode your model using information theory, which was quite slightly. And again, all of these tools are available in R, and actually it's not that much code. I'm actually being quite verbose, and I still only have roughly 50 lines of code. Okay, so I think that was it for the R Studio part. So I will just go back to the presentation, sharing the presentation here. One moment, sorry. It is here, share, and go to full screen here. So let's wrap up. Now we've sort of been over the theory I showed you in R, and we are running out of time. but I still think that we did quite nicely. So let's just briefly talk about this post hoc analysis and see if it makes sense. One thing is conducting your post hoc analysis, but be sure to actually check that it makes sense. In my case, I want to map it to some biology, since I'm working with biology, more over immunology. So this was the logo that we got out from the strong binders. Look at the very strong hydrophobic signal in the 2nd and the 9th position. Let's compare this to an actual structure. Long story short, this is a cartoon figure of how a protein would actually look. The brownish area here is the MHC. The sticks here in the middle is the peptide. So what you can actually see is that there's sort of stuff from the peptide protruding, sticking out of the MHC, and the stuff sticking into the MHC. And if you look closer into the details, you can actually see that it is exactly the 2nd and the 9th position that are sticking into the MHC, meaning that they're important for binding. So this is mapping your post hoc analysis, information theory based deconvolution of what the network actually worked to your actually biological problem. Remember, know your data, understand that the domain-specific knowledge is really important. So very briefly on model deployment, if anyone is listening and working in companies where you need to deploy a model, all of this is possible using this TensorFlow. Basically, you have what's called TF deploy package. And what you can do is that you put a REST interface on top, and then you can serve your models directly. And you can actually, when you do the serving, it's compiled down to just the C++. So you don't need all the R and Python runtimes. And this really means that you bring together the best of all the worlds. You have R, which is phenomenal for exploratory data analysis, and training and authoring on all these models. And then you can deploy, uses the languages which are well suited for deployment. You can even deploy using JavaScript directly in the browser or directly on the user client-side mobile phone, for example. So here are just some listed of the suite of the TensorFlow R packages, the APIs, there's some TensorFlow estimators, TensorFlow itself, TF dataset, and there's a bunch of supporting tools that you can check out if you're interested. CloudML is quite cool if you want to work on Google's cloud engines. And then you have various resources. I mentioned briefly this deep learning for R book by Francois Joulet and Gigi Allaire. That's really quite cool. R for data science by Grolman and Wickham. Super good book for learning the modern data science. And then there's this deep learning book that goes more into sort of the details, which is all quite cool. A lot of online stuff that's available. And I know that I know I keep mentioning RStudio, and it's not because I have any affiliation with them. I'm really happy about how they're contributing to the community. And of course, they have a business plan, but quite a substantial amount of their revenue actually goes into creating open source software that we can use in academia and in teaching, which is very, very cool. And it's really about sort of making the technology available. The technology, it might be cool, it might be fantastic, but if it's not available, it's going to remain unused. So in summary, the paramount criteria for any technology, if it wants an impact, is it has to be available. And with the TensorFlow, we really sort of see a revolution in application of these neural networks. And the Keras API really enables this fast implementation. Again, being able to go from idea to result with the least possible delay is key to doing good research. It's a bit of success. It's really important to understand why that is sort of the paramount way of doing your research. We're basically iterating over ideas. The more ideas you can iterate over, the more likely you are to stumble on the right idea. So this really means that model building ends up being not as much about actually doing the code, but more about mapping your problem to the data and then finding a good solution. So I showed you the code here, and it's not super hard code to write. The hard part is really to figure out how do you build your model, which architecture should you choose, and how do you choose which hyperparameters to include. And that's really where a lot of this time is spent. And really in this course, in this webinar, that's really a case in point, right? In this talk, we spent relatively little time coding and much more time understanding. And that's really an important point I would like to make, that understanding your problem, understanding your data, understanding the technology that you use, that's what you actually is preceding the good solution to your challenge. And just a final remark about architecture and hyperparameters. I always get asked about this. How do you choose the best hyperparameters? There is no clear answer. It's a lot of time. And basically, you should really be aware that in hyperparameter space, no one can hear you scream. And without further ado, thank you very much for your invitation. I went slightly over time. I hope that's okay, and that you are still here. Thank you very much. I will be happy to take questions now.
So, Leon, thank you very much for this talk. Insightful as usual. I must admit, I loved especially the Lego analogies. You can clearly see that you are that this talk that the talk is given by a Dane when you see Lego, one of the best toys ever.
True.
Oh, maybe we'll get a sponsorship. Who knows?
Yeah.
But still, going back to the serious questions, we will start with some questions about serious bioinformatics
Nice.
from I suppose Broad Institute. So epitope binding doesn't always activate T cells, and output signals could be really noisy. And how do we how do we take care about it during the transformation? So this is exactly a weak supervision problem, a problem that we have a lot of noise in the labels. So how to solve this? How to address this issue in your model, Leon?
So first of all, let me break it down in two parts. It's true that epitope binding, meaning actually peptide binding, doesn't always activate the T cells. Remember that if we are supposed to end up with an epitope, it needs to be able to activate the T cell. But you can have a peptide which is actually a foreign peptide presented on MSC and still does not activate the T cell. So why does this happen? It happens because there's something called central tolerance. Basically, T cells are not allowed to get activated upon seeing self-peptides. This means that if there is a high degree of similarity between the self peptide that the T cell has been trained on, so on, and the non-self peptide from the virus, you'll end up in a situation where you actually have a virus peptide, but it doesn't activate. So the final piece of this puzzle is on the T cell side, and understanding how the T cell recognizes this PMHC is really non-trivial, and it's the main area of my research. With respect to having noisy data, we can actually get quite clear data. Of course, there's always noise in assays, but for especially the tetramer assays for the determining MHC binders, they're quite accurate. And we're working together with Eyrich Jessen's lab at DTU, and they're creating this amazing barcode technology where you get a very clear signal, and you can even multiplex a lot of things together. So this is really the foundation of the training data. And actually, a lot of data is available. If you go to if you look up the Immunology database, you'll find a lot of the data. I hope that was a sufficient answer.
Thank you. So the next question is also hardcore bioinformatics. We know for a fact that affinity distributions differ between the classes of the binders. So low, medium, and high binders have a very significantly different affinity distribution. And so are we simply modeling the affinity using the weights?
So actually what we're doing is that in the predictors that we have in our lab, they are trained on two types of data. So they're trained on affinity data and they're trained on what's called eluded ligands. In the example I showed here, it's simplified. So I trichotomized a continuous score to sort of make the example more digestible, basically. But the way that these peptides are scored is that they are scored as a percent rank score, it's called. And basically it means that roughly somewhere between 1 in 200 to 1 in 500 peptides will be presented by the MHC. So the idea is if you take the full-length peptide and you predict on all, the full-length protein, and you predict on all the peptides, and then you sort them according to the score, are we then able to actually put the correct peptide way towards the top? And we are. And that's really the point here. And also there's this thing about using binding affinity as a cutoff. We can see that across different alleles, different thresholds of binding affinity defines binders. So with respect to modeling affinity over the weights, we are modeling a combination of the affinity and the eluted ligands. We have actually a network which is trained on both. And then the idea is that if you have an eluted ligand, so you take a cell and then you lyse the cell, you capture the MHCs, then you elute the peptides, and then you know which peptides were bound to the MHCs. This is your data, so that's binary, bound yes, no, whereas the binding affinity is a continuum. Then you have a network with two outputs, and then basically you train the network towards each of these neurons, and then you backpropagate across both sets of weights. So basically sort of transferring learning between the data point. I hope that was helpful.
Okay, so the last of the bioinformatics questions from a viewer who is captivated by the context of amino acids. So you were during the presentation, you were showing us that amino acids, it's not only about which amino acid, but also in which context is it presented. So do these measures for amino acid similarities take into account context of the context in the peptides? And could you say a bit more about these measures?
Okay, so the one that I showed you was the Blossom 62 matrix. That was the one that we used. I put a link in the cloud workspace, which I'll make available. But very briefly, basically, the idea is that you take evolution as a point of reference. So you take a lot of proteins which are basically the same family. Blossom is the block substitution matrix. This is the blocks database. And then per position, you see how often is an alanine exchanged for a leucine, for example. And that gives you an indication of the viability of such an exchange, meaning that if that's a very radical change, the protein would not survive. I mean, there would be an impact so that the species would be affected by this, and most likely the protein would not survive in an evolutionary context, meaning that there's a big biochemical difference. It does not take into sort of this single score does not take into account in which context that particular score was observed. But what you can actually do is that we know the biochemical composition or the biochemical profile of each of these amino acids, be that positively, negatively charged, or whatever. So you can actually take the Blossom matrix, and then you can do a principal component analysis, and then you can project the amino acids into 2 dimensions. And if you do that little exercise, it's actually quite fun. Then you see that you do in fact get the charged amino acids together, you get the hydrophobic together, and so on and so forth. This means that the block substitution matrix, which is a set of log-odd scores basically, is actually a proxy for the biochemical uniqueness of each amino acid, because in evolutionary context, it defines whether or not a certain substitution is allowed, quote unquote.
Thank you. It's always good to have a bit of this. When you're doing the bioinformatics, it's always good to have in mind a bit of evolution and how basically it's all about it's all about chemistry at the very end and the properties of amino acids. And it's really great that such properties we can model with neural networks. And then right now we have a bit of questions a bit of questions from our viewers who are curious about the technical part of your talk. So first and for all, as a user, could you recommend RStudio Clouds for for computing intense tasks. So would it handle the need of 64 gigabytes of RAM and 8 CPUs, or is it more regular? And also from could you efficiently use GPU using RStudio Cloud?
So as far as I know, there are no GPUs on RStudio Cloud, as far as I know. But I know that they recently implemented a payment plan, meaning, among other things, that the free version, which was the one I was using, it has 1 gigabyte of RAM, which is not a lot. It has 1 CPU, and you can only use it for 15 hours per month. So no, I would not recommend RStudio Cloud for heavy computations, or at least not the free version of it. I don't know if they have a payment plan where you can really max out your resources, but I do know that, for example, Amazon Web Services allows you to install RStudio Server, and then there you can manage your own resources. Of course, you pay for it, but if you want to do heavy computations, heavy neural networks, then you need GPUs. Very quickly, with more complicated networks, the training time gets very, very high. And this means that you're no longer able to iterate over ideas quickly, because each time you get an idea, it may take as much as a week to actually figure out whether that went well or not. So it's really a matter of finding access to the right resources if you want to do efficient research.
So the next question comes about the reproducibility and the usability of the whole thingy, and by thingy I mean Keras R API, because we have a lot of layers. So we have R, which is a wrapper around Python code, which is a wrapper around C++. So how to ensure reproducibility in such environment? And reproducibility on many levels, because we are starting with the level of software. So we need to version software and so on, but also we are doing the neural network. So we want to keep seeds, and we want to, for example, promote our seeds from the highest, from the control, the random seeds from the highest level to the.
Yeah, so there are several levels in this. So basically, with respect to handling the reproducibility of the software itself, that's an entire talk in itself. I know that there's Will Landau did this Drake package, which is quite cool. I know that some people are using Docker containers to maintain the software versioning. So that's that's one side of it. The other side is exactly this: if there's a if in that there's a degree of randomness in your initialization of your weights and so on, you will get slightly different results when you run your networks. You can actually, but there's a caveat to this, you can actually set a seed which works down through the call stack. However, as far as I understand, this only works if you're using CPUs. It doesn't work if you're using GPUs. There's probably a technical reason for that. I'm not aware of exactly how that works.
Okay, and we have a follow-up question about the reproducibility issue.
Mm-hmm.
So is usage of ensemble of neural networks improving the output predictions?
Yes. So actually, what I showed you today was just, again, a brief example. What we would usually do is that we would use five-fold nested cross-validation. And without going into too many details, basically you end up with 20 models. And then you have 20 models with an ensemble of neural networks. And then if you want to predict on a new data point, you predict on network 1, 2, 3, 4, all the way up to 20. And then the output of that final prediction is the average of these predictions. And we have shown again and again and again that this is really powerful. It's sort of the wisdom of the crowd approach. And ensemble networks, they're a bit more cumbersome to handle technically. But with respect to actually getting more well-performing, more robust, less overfitted models, they actually seem to work quite nicely.
Thank you very much. And just to wrap up the Q&A with some soft question. So Leon, deep learning and R in the consciousness of, let's say, imaginary and average data scientist, these two things are not really merging together. Why are you using deep learning with R? What is the reason behind this choice? Is R more convenient for deep learning in bioinformatics?
So why am I using R? So I really like using R, and especially with the advent of tidyverse, which I really got into. I think that it's really powerful, and it allows me to quickly do the exploratory data analysis I need to do. It allows me to do the wrangling, and also the whole ggplot allows me to do the visualization. So that's sort of an ecosystem that works really, really well. And then there's the added layer of wanting to do machine learning. And there's this, I'm not entirely sure why, but there was a thread on our studio community the other day about why R is failing in machine learning. And I don't agree that it is. I think it's a matter of what's being promoted and what's being used. There's Bernd Bischel's MLR group, for example. They're doing amazing stuff. Then there's what's his name, Max Kuhn, with the whole first the carrot and parsnip and tidy models. That's also really cool. There's Ranger. Was it Marvin? What's his name? Yeah, and all
Marvin Wright.
Yeah, that's the one, exactly. Marvin Wright is. All these things, they're doing amazing, and it works really well. But there's just sort of this. I think that it sort of boils down to the fact that R was classically developed for classic statistical analysis, and this is sort of still its strength. And then people, if they want to do learning, then they go to Python. Having said that, I think that it's important that we move away. I really highly, warmly feel that life is too short for language wars. It's too short for text editor wars. We should work towards getting a unification of data science, and we should work towards getting a language agnostic way of incorporating the different things that we can bring to the table. So, for example, you have Hadley Wickham, the whole sort of main guy behind tidyverse, and you have Wes McKinney, who's behind pandas if you're using Python. So pandas and the way of handling data frames in tidyverse are very similar. And I know that they are brewing on something together. So I'm quite interested in seeing what this works out. And I actually also know that the RStudio editor itself has support for being used with Python. You can even mix Python and R together. So you have a script which uses both languages at once, and you can interchange the objects, which are quite cool. So why I'm using R is for deep learning is that I get the entire ecosystem. I get the handling of the data, I get the visualization of the data, and I get the modeling of the data. And I like that, and it works for me. I have no problem with people using other tools at all. I think it's important that you use the right tool for the right task.
So, Leon, thank you very much. And just a few finishing words. So, Leon is a great proponent of open science. So, of course, you will be able to find his talk, link to his talk and his code after in the we'll put these links into the description of the video.
Absolutely.
So to all of you who wants to redo the analysis on your own, it will be available for you. And once again, Leon, thank you very much for spending your time on our webinar, for showing us all those great things, for priming the bioinformatics topics on our webinar series. It's really amazing. Big thanks to our viewers and for your kind support. Your every like, every share means a lot to us. It allows us to get to share our passion for R, for data science with much larger crowd. So thank you very much for being here, and good night for everyone in the European time zone. Bye bye.
Thank you. Thank you.